Good evening, this is Mr. Wintermelon. So in the previous chapter of cell division, we have learned about mitosis and meiosis. Meiosis is the process whereby sex gametes are produced. The sperm and the egg contains information which is passed down from our parents to us. And we will learn more about them in this chapter of inheritance. So, have you ever wondered how come you resemble your father or mother? Or how come your neighbor Tom, Dick and Harry resemble the milkman more than their father? Or have you ever wondered how come the cup looks like the lion instead of a sponge? And have you ever encountered this question being thrown to you? If evolution is true, how come the monkey, okay, just take note, it's a chimpanzee, but most of the time people will say monkey, in the zoo have not evolved into a person yet? So all of this will be answered in this series of lessons. Just a brief chapter overview. First, in this video, we are going to learn about the lingo of genetic, which is a fancy way of saying the terminology involved in this chapter. Next, we are going to learn about monohybrid crosses. Do not be daunted by this. They are actually very easy. Then we are going to learn about causes of mutation and specific example. Then we are going to talk about evolution in the last lesson. So let's start with some of the words that you have to know. So by the end of this lesson, you should be able to distinguish between a gene and an allele, explain what does some of the terms means, as well as to describe the difference between continuous and discontinuous variation, and give some example of each. First, take a look at the photo of these 6 puppies. Which one is the odd one out? Clearly, it is the puppy in white. Even though these 6 puppies were from the same parents, that means they received similar genetic material from the parents. But how come the appearance of this white puppy is so different from its siblings? To understand this, we need to know what is the difference between genes and traits. Genes is the instruction for making protein, and the genetic makeup of a person or a puppy is known as their genotype. And the genotype of a person will affect his or her traits. Traits is another word for quote-unquote appearance. There are inverted commas there because it doesn't have to be things on the outside. For example, if a person is allergic to peanuts, you can't tell from the person appearance or from the outside that the person is allergic to peanuts. But nevertheless, it is still the trait of a person. And we use the term phenotype to describe the traits. So your genotype will affect your phenotype. Your gene will affect your traits. So to put them in words, genotype is the genetic makeup of an organism. They are inherited from our parents. The genotype will lead to phenotype. Phenotype is the trait expressed by an organism. Different genotype can have the same phenotype, and your phenotype can be affected by the environment. For example, your height and weight. If you eat a lot more calories than you are burning off, you will most likely get fat. So this is what it means by getting affected by the environment. And the differences in the phenotype of the same species will result in variation. What is variation? Variation is the differences in the traits between individuals of the same species. And you might want to copy this down, as well as the two types of variations that we have. First, we have the continuous variation. That means the variation is, continuous. It has a range. For example, your height and weight. So we have people whose height is 140 and maybe all the way up to 2 meter plus. There will be height in between, for example, 161.2, 161.3, 161.4 cm, and so on. It is continuous. There is a range. Continuous variation is often controlled by more than one genes, so those genes have additive effect. For example, someone that have many copies of the genes that produce the black pigment protein will most likely be more tan or darker than someone with lesser copy of the genes. And continuous variation is most of the time affected by the environment condition. For example, your height and weight, the amount of nutrients you get, especially during your puberty, will affect your height and weight. Besides continuous variation, there is also discontinuous variation. They are discrete phenotype whereby the difference is distinct. What do I mean by this? For example, double eyelid compared to single eyelid. There is nothing in between, like 1.5 eyelid. Another example is whether you have the enzyme to digest alcohol. It's either you have or you do not have it. So even if you drink a lot of alcohol, increase your alcohol tolerance, your face will still become red. So normally this continuous variation is not affected by environmental factor. So it's controlled by one or just a few genes, and there is no additive effect. Just take note, there are some variation that can be both continuous and discontinuous variation. For example, your skin color is kind of continuous and discontinuous. So what do I mean by this? It is quite obvious that there are different shades of skin tone, like maybe 50 shades of brown, 50 shades of orange. It really depends on how much sun you expose yourself to. But nevertheless, they are still kind of discontinuous. For example, you can kind of tell a white and a black guy apart. So while there is different shades in individual skin color, but comparing one skin color to another distinct skin color, for example white and black, it will become a discontinuous variation. So just bear this in mind. And this variation is controlled by our genes. So our gene will control whether we have single or double eyelid, whether we have curly or straight hair. This is a discontinuous variation. Either our hair is curly or they are straight naturally. Of course, you can perm or go and reborn. But naturally, there are curly hair versus straight hair. So these are just traits on the outside that we can see. But there are also phenotype that is sort of on the inside. For example, whether a person can drink milk or not. A person who cannot drink milk, we call them lactose intolerance. And as mentioned before, whether we have the enzyme to break down alcohol. If we do not have it, it will cause our face to become red. So our gene will affect our phenotype. So different phenotype will definitely have different genotype. But does the same phenotype have the same genotype? That means if a person has straight hair, does it mean that he or she would have the same genotype as another person with straight hair? Not quite. But to understand this, we need to first learn a few new words. Genes is something that we have learned in the previous chapter. It is a unit of inheritance, a small sequence of DNA in a chromosome that codes for a protein or control a particular characteristic or traits. Alleles were briefly mentioned. Alleles are different forms of the same gene that occupy the corresponding position on homologous chromosomes. For example, we have a pair of homologous chromosomes, each with 4 genes. So we have the gene A, gene B, gene C, and gene D. Now, you will notice that some of the letters are capital while some of the letters are in small cases. That is because we use capital letter and small letter to, or at least this is what we do most of the time, with the exception of blood group, which we will go to in the next lesson. Anyhow, for alleles with capital letter, they are known as the dominant allele, while the small letter is known as the recessive alleles. So alleles actually have two different forms, the dominant form and the recessive form. And we use two words to describe the different combination: homozygous and heterozygous. Homozygous means that both alleles are the same, while heterozygous means that both alleles are different. Let me give you an example. Gene A is homologous dominant because both the alleles are the dominant alleles. As for gene B, we say that they are homologous recessive because the version of gene B are the recessive alleles. For gene C and gene D, we say that the chromosome is heterozygous for both gene C as well as gene D. So for heterozygous, it's pretty straightforward. As long as one is the dominant alleles and one is the recessive allele, we will call them heterozygous. For homozygous, we can use it most of the time, but if we want to be specific, we can say that they are either homologous dominant, whereby both the chromosomes have the dominant alleles, or we can say that it is homologous recessive, whereby the chromosomes have both recessive alleles. So what's the consequences of inheriting a dominant or a recessive alleles? For dominant alleles, it will express itself to give the same phenotype in both homozygous as well as heterozygous condition. This is a fancy way of saying that you just need one copy of this allele for you to have the phenotype which the alleles is supposed to give. While in the case of recessive alleles, it will only express itself in homozygous condition. The alleles does not express itself in heterozygous condition. To give an example, let's use the enzyme that breaks down alcohol. As we all know, genes is a sequence of DNA that codes for protein. And your enzyme is made up of protein. So if a cell have the dominant alleles, it means the cell has the instruction to produce the enzyme. And as long as your body cell have one copy of this dominant alleles, it will have the required information or instruction to produce the enzyme that breaks down alcohol. It doesn't matter if the other alleles is recessive or dominant. A good analogy would be you just need one eye to see. It doesn't matter whether the other eye is blind or not. As long as you have one working eye, you can see. In the case of recessive alleles, the instruction to produce the enzyme is different from the dominant alleles, which is why it is different form of the same gene. And because the instruction is something different, the protein produced might not be a functioning enzyme. So both the homologous chromosome needs to have the recessive alleles. That means both the homologous chromosome need to have the sort of defective instruction to produce the enzyme for the person to not be able to produce the enzyme, and therefore will express the recessive alleles phenotype, which is unable to break down the alcohol as quickly as someone with one or two copy of the dominant alleles. So he or she, her face will turn red rather quickly if they consume alcohol. So this is the difference between dominant alleles and recessive alleles. Dominant alleles normally produce a working protein, while recessive alleles normally doesn't produce a working protein. Having said all of this, you might get the misconception that dominant alleles is better. But let us look at two case studies. The first case study I would like to look at is some genetic disorder. Genetic disorder are diseases caused by specific alleles. There are two classifications of genetic disorder. First is disease caused by dominant alleles, as well as disease caused by recessive alleles. This disease, called the Huntington disease, is actually caused by a dominant alleles. That means the person will just need one copy of this allele to get this disease. In the case of cystic fibrosis, the patient actually have two copies of these recessive alleles because it is a disease caused by recessive alleles. Hence, the patient have to be homozygous to get the disease. The important thing to take note is just because a gene is dominant doesn't mean that it produces something good. Because for the case of Huntington disease, the dominant alleles actually cause the condition to develop. For the second case study, I would like to look at our blood group. If you can recall from the chapter of transport in human, we have 4 blood groups: blood group A, blood group B, blood group AB, as well as blood group O. Blood group O is the only blood group without any antigen. That means the alleles actually doesn't have the information to produce either the A or B antigen. Therefore, blood group O is the recessive alleles, while blood group A as well as blood group B, they are the dominant alleles when we compare to blood group O. The case of blood AB is a bit special. It is known as co-dominant, and we will discuss more about this in the future lesson. So let us just keep it as it is. Group A and B are the dominant alleles, and group O is the recessive alleles. Now, let us look at the blood group distribution in Singapore. Which of the blood group do you think is most common in Singapore? Make a guess. So the most common blood group, as you can see, is the one in orange or dark orange or brown. It is actually blood group O. Is this only unique in Singapore? Let us look at the distribution around the world. So around the world, even though blood group O is slightly less than the 45% in Singapore, it is still the most common blood group out of the 4. So just because O allele is recessive doesn't mean that blood group O phenotype is the least, as we can see from the pie chart. So this is an important point to take note. Just because something is dominant doesn't mean most of the people have it. Just because something is recessive doesn't mean that it have the least amount of phenotype in a population. So the take-home message for dominant alleles is this: just because an allele is dominant does not mean that it is a good phenotype. And just because it is dominant doesn't mean most of us have it, in the case of blood group, as well as many other examples. Especially in the case of disease, most of us do not have Huntington disease. That means it is actually rare to have that dominant alleles, since you only need one copy for the person to develop the Huntington disease. So dominant alleles just means that it will express its phenotype in both homozygous and heterozygous condition. It just needs one copy to express its phenotype. Nothing good, nothing bad about it. It is very neutral. And one last thing that I want to highlight is, remember prophase 1 of 没有事没有事没有事. What is the important event that took place during prophase 1? So we have our homologous chromosome, and let's say the crossing over takes place, and gene D actually switch between each of the homologous chromosome. And this is how the homologous chromosome will look after crossing over. You can see that the combination of alleles is now different. Originally, the homologous chromosome on the left have 2 dominant alleles, but now it only have 1. Whereas the homologous chromosome on the right, originally it have 2 as well, but now it have 3 dominant alleles. So now with a better understanding of alleles, I hope you can appreciate why crossing over during prophase 1 of 没有事 meiosis can lead to genetic variation.